Question number 33. Question number 33. For each molecule of ammonia produced by nitrogenase enzyme, the number of ATP molecules are required during the reduction of nitrogen. During the reduction of nitrogen, that is 2 nitrogen and 2 dinitrogen is required. Then 8 protons are needed for this one, that is because it's a reduction process, plus 8 electrons are required. 8 electrons are required and 16 ATP. Here 16 ATP are required by 2 nitrogen. Results in the formation of 2 ammonia. Results in the formation of 2 ammonia plus 2H, that is H2 or 2H, better to say H2, then 16 ADP and 16 phosphates are released, inorganic phosphates. So see here, nitrogen reduction is an active process. Nitrogen reduction is an active process, means energy is required, and for 2 nitrogen 16 ATP are required and 2 ammonia molecules are formed. But see the question here, each molecule of ammonia. Question says each molecule of ammonia. For the formation of 2 molecules of ammonia, 16 ATP are required. So that's why the answer is 16 by 2 for 2 ammonia, that is for 1 ammonia. Answer 8. Answer 8. So 33rd question, answer 8 for the formation of 1 ammonia. For the formation of 1 ammonia, 8 ATPs are required, and any process in which ATPs are needed is termed as active process or energy consuming process. Reductive amination. Now come to this activity, reductive amination. Reductive amination is actually the alpha ketoglutaric acid. Alpha ketoglutaric acid is already present in the cell, that is available from respiration. Alpha ketoglutaric acid. This alpha ketoglutaric acid reacts with ammonium ions, reacts with ammonium in presence of enzyme glutamate dehydrogenase. In presence of enzyme glutamate dehydrogenase, and forms glutamate amino acid. Glutamate amino acid. This is the main amino acid. This is the main amino acid. So see the answer. See the answer. Reductive amination, that is inclusion of ammonium ion with the alpha ketoglutaric acid. Inclusion of ammonium ions or combining of the ammonium ions with the glutamic acid in presence of an hydrogen donor, in presence of a hydrogen donor. Here hydrogen donor is also required. Hydrogen donor is also required here, and hydrogen donor is NADPH. NADPH. Hydrogen donor is NADPH. Donates hydrogen for this activity. Addition of hydrogen is reduction. Addition of hydrogen is reduction. So see this one, that is in reductive amination, alpha ketoglutarate reacts with the ammonia, better to say ammonium, and form what? That is glutamate. And what is formed? Glutamate is formed. Glutamate is formed. So answer number 2 is correct for this reaction. Answer number 2 is correct for this reaction. Which is the following an enzyme required for the reductive amination? I have already named it glutamate dehydrogenase in the previous reaction. That is alpha ketoglutaric acid plus ammonia plus plus hydrogen donor in presence of glutamate dehydrogenase enzyme. What is formed? Glutamate is formed. What is formed? Glutamate is the main amino acid produced in this process. The main amino acid, again that question is also linked with the previous one, that is answer glutamate. Question number 37. Most important amides. One is asparagine and 2 is glutamine. That's why answer is 1 and 2. Amides are more stable than amino acids. Amides are more stable than amino acids. In amino acid, there is a amino group, that is NH2. This is alkyl group. This is COOH group. If this is removed by ammonia, this is again removed by the amino group, then amides are formed. And amides are more stable than amino acids, so they can easily be translocated. They can easily be translocated within the plant. Nodules of some plants export the fixed nitrogen as, yes, in soybean urites. Soybean, this answer for this question is soybean and urites. This is applicable for the soybean only, not amide, only urites, and that too is in soybean. Leg hemoglobin, a pink colored pigment. Here the leg indicates concern with the legumes. Leg for legume. Leg for legume because it is found in nodules of leguminous plant and they are pinkish due to presence of leg hemoglobin. And it is very similar to the hemoglobin, that's what is called leg hemoglobin. And nitrogen reduction process. N2 reduction process is actually anaerobic. N2 reduction process. Nitrogen reduction process is anaerobic. That activity is anaerobic. That is, oxygen is actually inhibitor for this activity, and that oxygen is trapped by leg hemoglobin. So that's why if the oxygen is there around the nodule, it is trapped by the leg hemoglobin. So it is needed for the removal of oxygen. It is needed for the removal of oxygen, or better to say it is an oxygen scavenger. Better to say it is oxygen scavenger. So question number 39, that is oxygen scavenger is leg hemoglobin in the root nodules of legume, a pink colored pigment. An enzyme playing important role in the nitrogen fixation. An element, sorry, not enzyme, in an element playing, that is molybdenum. Exclusive role, its exclusive role, molybdenum, because it is the structural part of two enzymes. One is nitrate reductase. One enzyme is nitrate reductase, sorry. Enzyme 1 is nitrate reductase. And enzyme number 2, nitrogenase. Enzyme number 2, nitrogenase. One is nitrate reductase, other one is nitrogenase. Nitrate reductase enzyme responsible for the reduction of nitrate into nitrite, that is NO2 level. Whereas nitrogenase enzyme is concerned with the reduction of ammonia into, reduction of nitrogen into ammonia. Reduction of nitrogen into ammonia. This is the role of nitrogenase, whereas nitrate reductase enzyme reduces nitrate into nitrite, and both the enzymes, they have molybdenum as a structural element. 41. Free living anaerobic. Look here the word anaerobic. Beijeringia aerobic, Rhodospirillum is anaerobic, Rhizobium is symbiotic. This is free living aerobic. This is free living aerobic. This is free living and aerobic. Whereas first one anaerobic. This is anaerobic. The question is anaerobic nitrogen fixer, and that is free living. That is free living. So my answer is Rhodospirillum. This is anaerobic. The last one is aerobic actually. Last one is aerobic. So this is correct answer, Rhodospirillum. Common nitrogen fixer in paddy field. Paddy field, the amount of enormous amount of nitrogen is needed, and azospirillum is found in the abundantly found in paddy field. Azospirillum. Rhizobium is symbiotic nitrogen fixing bacteria. Rhizobium is symbiotic. Rhizobium is symbiotic nitrogen fixing bacteria found in root nodules of legume. Found in root nodules of legumes. Whereas Oscillatoria blue-green algae, it is a free living. Frankia is found in root nodules. Frankia is found in root nodules of non-leguminous plant, non-legumes. Symbiont. The last one is symbiont. Found in Alnus, Myrica and Casuarina plant. Found in Alnus, Myrica, Casuarina. So answer, final answer. Question is paddy field. In paddy field actually azospirillum is abundantly found there. Symbiotic nitrogen fixer, here the answer is Frankia. Glomus, it is a fungi actually. It is a fungi associated with the roots of higher plant, and it is a part of mycorrhiza and no role in nitrogen fixation. Mycorrhiza, they are concerned with the phosphate absorption, phosphate nourishment. Azotobacter, free living aerobic bacteria. It is free living aerobic. And Frankia I have already described in the previous question, that is found in the non-legumes. Found in non-legume. Root nodules of non-leguminous plant. Root nodules of non-legume. Alnus, Myrica, Casuarina. So here symbiotic nitrogen fixer is R, and Frankia is symbiont. So answer is Frankia. Azolla is a pteridophyte. In this pteridophyte, a blue-green algae is present, Anabena. N-abinazole. N-abinazole. It is also symbiont, but azola can exist also as free living. Better one, symbiotic nitrogen fixer is Frankia. Ammonia is oxidized to nitrite during, if plant receives ammonia, sorry, not a plant, soil. If ammonia is there in the soil, first of all, it is oxidized into nitrite by nitrosomonas. So ammonia after oxidation forms nitrite first with the help of nitrosomonas. Then nitrobacter is also there. The further work is done by the nitrobacter bacteria, and they oxidize nitrite into nitrate. And both these, that is nitrosomonas and nitrobacter, nitrosomonas and nitrobacter, both are chemosynthetic bacteria. Both are chemosynthetic autotroph. Chemosynthetic autotrophs. Nitrosomonas number 1, nitrobacter number 2. These two are chemoautotroph, and they are also involved in the nitrogen metabolism. In that case, their role is to oxidize ammonia into nitrate, and this is two-step activity. Step number 1 is oxidation of ammonia into nitrite by the nitrosomonas, that is nitrosomonas is involved, and followed by the nitrobacter. So in this case the answer is nitrosomonas. The answer is nitrosomonas involved in the oxidation of ammonia into nitrite, that is NO2. First stable product of biological nitrogen fixation by rhizobium. Simple answer, that is ammonia. That is N2. It needs 8 protons and 8 electrons plus 16 ATP result formation of ammonia plus hydrogen, then 16 ADP plus 16 inorganic phosphate, 16 phosphates are released, they are inorganic phosphate. So this atmospheric nitrogen is converted into ammonia during nitrogen metabolism by the rhizobium in the root nodule of leguminous plant, and that entire activity is catalyzed by the enzyme nitrogenase. This entire activity is catalyzed by the enzyme nitrogenase. That's all for mineral nutrition. Thank you.